Kristen Barnwell.
I'm Kyle Bruce.
And our advisor is Dr. Coglin. And our project is on research on tardigrade radiation resistance for applications in space exploration.
The reason we focus on tardigrades specifically is because they're kind of an extremophile, so they're able to survive in pretty harsh conditions. One specific condition that we're looking at is the fact that they're pretty resistant to radiation. They can stand up to 5,000 grays of radiation, whereas a human typically dies within the 30 range. So, space radiation, what is it? So on the electromagnetic spectrum, practically once you get into space, anything above visible starts to become a problem because you're not within Earth's kind of protective realm. The wavelength gets smaller, so it's able to penetrate further.
And then obviously this is dangerous because the effects that it can have on somebody traveling in space, there's acute effects such as nausea and vomiting, and then it can go as crazy as changing your DNA and central nervous system, and that'll heighten your risk of cancer. So obviously that's not good. And that's going to limit our exploration abilities just because we don't want to send them out. You can't have plants to grow for food and things like that. Our objective for this is to identify factors that contribute to radiation resistance, with a focus on tardigrade molecular makeup and determine applicable nanotechnology for space exploration.
So one way we're going to do this is that the tardigrade kind of has a couple things about it. So we're specifically we've identified trehalose. I might be mispronouncing that, but it's a molecule within the tardigrade that researchers believe causes the resistance to space or to DNA repair. What it does is, I believe it's also something that's related to anhydribiosis or something. It's about whenever it gets desiccated, so it dries out. It's the molecule that's responsible for that. And they think that the function in which this can dry out or kind of protect DNA against dehydration also kind of helps protect it against ionizing radiation. So our hypothesis is that trehalose repairs the DNA of a cell affected by UV radiation. Our methodology, we're going to cultivate some. There's some techniques we found online that researchers have published about producing a yield of trehalose. We'd like to apply it to a DNA medium, and then we'll have a control group that doesn't have it with it, one that does have it with it, and then we'll subject it to ultraviolet radiation, and then measure the probability of whether one or the other grows better, or if the trehalose helps protect against it. And then from there we can move forward in terms of looking at the application of nanodelivery. So there's the far-fetched, or not far-fetched, but kind of further along one, which is applications to human DNA, but then we can also, on the short term, look into producing food in space without it having to worry about radiation.
For industry applications, obviously it's the main ones you think of that are sending stuff into space, like NASA, SpaceX, all the ones up there. But it's also, NanoRacks is the ones that help send little kids experiments up to the ISS and things like that. And TARDIS is a whole different experiment that they had. It's Tardigrades in space, where they had a group that was subjected to a space vacuum and heat radiation, and one just to the vacuum to see the different changes they had when they developed and reproduced. But overall, NASA has a lot of things that have different spin-offs, such as invisible braces and artificial limbs. So in the end, it affects all of us. So
this is a nice quote by Elon Musk. Kind of drives the point home in terms of spacefaring civilization. Here are references. Any questions?
All right. So tardigrade. Okay.
What's a tardigrade?
Otherwise known as a moss piglet or water bear. It's a very small little, animal that can survive in really extreme conditions. I guess would be the easiest way to explain it.
So are you going to test your tardigrade as well, or just directly into the DNA?
We can. I think we can kind of mimic or expand on the trehalose, where if we find other things in literature that people might be saying is a different mechanism in which the tardigrade uses, we can try to use those things as well in parallel with the trehalose. But at this point, I think we're mostly identifying the compounds or the molecular makeup within the.
Sure. So is your trehalose that you cultivate, what's the source of that?
I think it's like yeast.
So you will get the trehalose that's produced by another organism.
Correct.
And you're gonna try that and see.
Yeah, we're gonna try to find some sort of. We're still in the preliminary phase where we're trying to find the medium that we can apply to. Like I don't know if there's some sort of algae or something that might be able to that we can by some mechanism attach it to
Deliver it.
or deliver it.
Yeah, diatom is your friend there. But yeah, I'm just curious of the trehalose. There's no synthetic trehalose available yet that is produced, or they're basically really getting the yeast to produce that, or is it an
I'm not sure if there's an artificially
not
made,
yet? Okay.
but I know that from what I read, it was kind of like an expensive process to produce them, and then there was a recent kind of breakthrough.
And that's what you're testing, basically, and see what happens.
Yes, yeah.
That's awesome. You think you can do that in the semester?
I think we start. Yeah.
He'll try. Dr. Coglin's gonna open his lab for that. Okay. Any other questions, you guys? Okay. We'll wait. We'll see where the tardigrade comes.